So good evening, everyone. I thought I would do this in two parts. It's a short introduction. I'll tell you a little bit about the Society for those of you who don't know about us, and then I'll tell you about our lecturer. So as a learned society, we're now young for some societies, like the Royal Society, but we're over 85 years old. We're a modern society. We've done quite a lot to change ourselves over the last few years to make ourselves really fit for purpose for the next century. We have three journals: the British Journal of Pharmacology, the British Journal of Clinical Pharmacology, and a relatively new journal, which is Pharmacology Research and Perspectives. We run that one with the American Society aspect. We run scientific meetings. We have an annual scientific meeting, usually run in London in December. That collects around 1,000 people from, last time, over 50 countries. And we have guest societies join us last year. It was 2 American societies and the Chinese Society of Pharmacology. Next year we have the Japanese Society. So we have an international perspective at our meetings. We also run smaller conferences, workshops, Gordon Conference type things, Keystone conferences in the UK and overseas. And we support the International Union of Pharmacology, and we support the European Pharmacology Society and Clinical Pharmacology Societies. And we will be running the World Congress for Pharmacology in 2022, which is the one after the next one in Japan. We have a major focus on education. We've developed a new curriculum for pharmacology to try and make it fit for modern careers in the pharmaceutical industry and elsewhere. We've developed with the Medical Schools Council a prescribing safety assessment test, which is now taken by all 8,000 medical students in the UK and has to be passed before they can enter their core medical training. That's now a requirement. So it helps medical students focus their minds on prescribing and hopefully makes them better prescribers as a result. Since the last 2 years when I've been president, we've had a major focus on policy. We've always been we've always cared about policy, but we're really doing quite a lot of policy work at the moment. And the sort of things we've been involved in is work on Brexit, Lord Stern's review of the Research Excellence Framework, looking at banned chemicals and the problems they're causing for pharmaceutical development and for research in the universities, looking at performance and image-enhancing drugs and the cost and affordability of medicines. We've also been fighting with Munir Pir Mohammed as a lead to make sure that clinical pharmacology has a future in the UK. It's always been under some strain, but we're working very hard to make it increasingly successful. We give grants and awards to our members and fellows, amounting to the order of £300,000 each year, bursaries to travel to meetings, an important part of what we support. We're based in the UK. Our name is the British Pharmacological Society, but over 20% of our members are overseas, and we have 35% of our members are under the age of 30, and around the same amount are women. So we have a diverse organisation. It's become our custom, I guess, over the last 5 or 6 years to host a President's Lecture in the summer, and I'm really delighted to host this year's lecture. A close friend and a great scientist, as I'm sure you'll see in a short time. Garret also, I think it's important to say, has been a great friend to British science over the years. He's based in the US, and I'll speak more about that, but he makes frequent journeys over to advise the British Heart Foundation, to advise the Wellcome Trust, has done work on the research assessment exercise. That must have been hard work, but very important for the future of our universities. So Garret is a great supporter of the UK. So Garret is the McNeil Professor of Translational Medicine and Therapeutics at the University of Pennsylvania in Philadelphia, where he chairs the Department of Systems Pharmacology and Translational Therapeutics, and he directs for, I think, 15 years or so, maybe, the Institute of Translational Medicine and Therapeutics. This is the organisation for translational medicine, and many of us have tried to emulate on a small scale what Garret does on a really large scale. His work's characterised by an integrative approach to elucidating mechanisms of drug action, drawing on work in cells, model organisms, and importantly, studies in humans. Garret's work has been fundamental to the development of low-dose aspirin as a treatment for myocardial infarction, one of the commonest treatments now used to protect patients with heart disease. His group also, importantly, was the first to predict and then mechanistically explain the hazards caused by NSAIDs, which should have been a warning to Merck. I know Garret shared his data with Merck, but they rather ignored it, and they ended up with rofecoxib losing its license. That's Vioxx, was the name that most people know it by, and subsequently withdrawn because of its actions on the heart. Garret's also discovered many products of lipid peroxidation and established their use as measures of oxidant stress in vivo. And his laboratory was the first to discover a molecular clock in the cardiovascular system, and he's studied the importance of these peripheral clocks and the regulation of cardiovascular and metabolic function. Garret's received numerous awards. I guess the Grand Prix Scientifique from the Institute of Paris, excuse my pronunciation, is probably the most important. It's a sort of cardiovascular Nobel Prize. And he also won the Schottenstein Prize, both in 2013. And rather closer to home, Garret was made an honorary Doctor of Science at the University of Edinburgh, and I think subsequently several other universities have done the same thing. And he was also the Cameron Prize winner at my university, which I'm very proud of. Shares that with Marie Curie and a lot of other rather well-known scientists. He's a member of the US National Academy of Medicine, an honorary member of the Royal Irish Academy, and a fellow of the American Academy of Arts and Sciences. And here in the UK, I'm delighted to say he was elected to the membership of the Royal Society in 2012. I'm delighted to welcome Garret to deliver his President's Lecture this year, which will be on drug development and translational science in the era of precision medicine. Thank you, Garret.
So thank you very much, David. It's a great honour for me to be here. I remember many frightening experiences presenting to the British Pharmacological Society when I was working here, and being chastened by the interrogation that I was subject to by people of our age sitting in the front row. But it's a great honour for me to be here to speak, particularly in this very storied institution. And I came up with this rather grandiose title, which actually probably more appropriately might have been termed Clinical Pharmacology 2017, because really the ideas behind translational science and its reduction to practice in what we call precision medicine, or aspire to as precision medicine, really had its foundations in clinical pharmacology. And it's a particular pleasure for me to see not only great exponents like David here, but many friends who practice this arcane art in the audience today. So I thought I might start by just describing what I mean when I say translational science. I mean the translation of basic discoveries to realize novel therapeutics, diagnostics, and indeed approaches to management that do actually benefit public health. So that's the framework that we're trying to address. So I was born in 1950, which seems like a long way back now. Really the number of new drugs that come to market, at least in the United States, have fluctuated up and down, but remained relatively constant over that period of time. We have moments of excitement like we did a couple of years ago. We have moments of depression like this one. But generally speaking, the number of new drugs coming to market since 1950 has remained relatively constant. And as we all know, what has really changed is the headline estimate of the cost of bringing drugs to market, which nowadays, according to some, is up to $4 billion. So the question that arises from time to time is whether this endeavor is indeed sustainable. And it's worthwhile thinking about where the system tends to break down. This nice piece of work shows the likelihood of moving to the next phase. So once you get into humans, you've got reasonable odds of making it to phase 2. But where things tend to break down is in phase 3. And over here there's an estimate of the likelihood of moving from phase 1 to launch, and it varies between disciplines, but in general terms it's around 10%. So we were in part motivated by this, because phase 2 has undergone a transformation from what it's supposed to be to what it is. So what phase 2 is supposed to be is where we elucidate the mechanism of drug action in human beings, and where we begin to parse the factors contributing to variability of drug response. And what phase 2 has become is an underpowered pseudo-efficacy study, which is a prelude to miscalculations that are reduced to practice in the design of phase 3 trials. And as we all know, in phase 3 is where most of the expense is incurred. Now, there are many disincentives to accumulation of the human capital relevant to the performance of what phase 2 should be. We've talked a little bit about clinical pharmacology being under siege. It's been under siege since as long as I've been in this discipline. And this slide illustrates another one of those disincentives, and that is that in the types of studies that one performs at that stage of drug development, the impact, and institutions in Europe seem to be obsessed with impact factors for some reason, but the impact of the journals in which this work is published is considerably less impressive than for basic discoveries, or indeed for those phase 3 trials that are actually successful, or even many of those that are not successful, that are published in much more impactful journals. This just illustrates, I think rather conservatively actually, the average cost of these trials at various stages of drug development, and is really just to bring to mind what we all know, and that is that most of the expenditure is in phase 3, so that errors in the design of phase 3 can be extraordinarily costly. And of course that figure of $4 billion incorporates the cost of failure. The cost of failure of a system that we know is inefficient. So one of the things I'll turn to is whether we can think of more efficient systems that aren't laden by the debt of that figure. So in the United States, and I think from talking to Munir this afternoon before this talk, in the UK as well, there's been a considerable erosion of confidence amongst those of us practicing science as our major funding agencies descend into the realm of casino conglomerates. The NIH is now moving to being a co-funder of science as opposed to a primary funder of science. And if your funding rates are around the 10th percentile and less, it really is extremely discouraging, particularly for investigators thinking of entering the fray. And of course that's what we can't measure, the people who chose not to enter when they were confronted with this type of reality. And this nice piece of work actually illustrates in the next couple of slides the problem, again, within the US. The ORO1 is the NIH grant for a principal investigator. It's the time you earn your spurs and you emerge as an independent investigator. And what we're looking at here is the median age of an ORO1 grantee. Even by my standards, that's quite elderly. So you can imagine that you're emerging as an independent investigator, and you're told you've got a pretty good chance to do this by around 45. So unless you have a serious personality disorder or an extraordinary dedication to science, this is a little bit of a disincentive. This is the age that people get their first ORA1, and here we're looking at the youngest 5%. So I got my first ORA1 in 1980 when I was 30, and you can see how things have deteriorated since then. So this is a real challenge. It's prompting the NIH to take a series of maneuvers in terms of softening the pay lines for beginning investigators for their first grant, but then they hit reality when they try to renew that first grant. So, and of course the knock-on effect of that is for those of us who are not writing our first grant, the pay lines deteriorate even further. And this slide from the same study just illustrates that the same pattern is evident whether the ORA1 grantees are performing clinical research or basic research. What we're looking at here is in the ascending line, the PIs who are over 55, and in the descending line, in each category, those who are younger than 55. So we have a real problem with human capital, certainly in the United States. And of course, you know, larger events in the world can mean that this could only be the beginning. This could even get worse. Not just in the United States, I might say. The unctuous Theresa May. All right. So prompted by some of those considerations, as David said, we founded an institute back in 2004. After about 2 years' worth of contemplation, we came up with a pronounceable acronym. We decided that we were going to focus in a particular area of science, that the institution was going to focus in a particular area of science. It wasn't that we weren't going to encourage people to do whatever they wanted to do, but that in terms of institutional resources, we were going to really concentrate them on a particular area of science. And this institute has now become a large one. It's a long-lived one. We support education. We support programs, both through top-down funding, but more importantly, from bottom-up funding calls. And in retrospect, one of the most important things to have done was to actually declare precisely what the objective of the Institute was at the time of its foundation. Because if you don't do that, particularly if you have a word like translational in the title, people will project onto you what they think you're trying to do and then attack you for it. So we had a very simple mission. We said we wanted to increase the number of investigators who had the capacity to perform their science in the space between proof of concept in cells and model systems and mechanistic elucidation of drug action, of physiology, of disease mechanisms in relatively small numbers of humans. And secondly, we wanted to try and identify and work to depress the barriers to their success. So a very simple but highly challenging objective, but one which put a focus on human capital for the reasons that I talked about before I came to this slide. Now, we were very conscious of the changes that were going on outside of our institution. So 20 years ago, the field was really dominated by large vertically integrated companies, which did everything from drug discovery through to phase 1, 2, 3, 4, through to drug approval. And really, in biotech, they were pretty good at identifying targets, maybe do a bit of proof of concept. And in academia, really, we didn't do very much at all. We discovered a lot of these targets that the biotech people took and ran with, but not very much. And of course, as we all know, what's happened over the last 20 years is there's been a progressive disintegration of these large vertically integrated companies, such that, it's not water, such that could be gin, such that that now the modules of the process can be drawn from any of these sectors, including academia. They can be drawn from different geographies. And ideally, that's exactly what you would want, because the best people in these different stages of this very variegate process in different fields, no matter how large the company is, they're not going to be within house. They're going to be scattered all over the world in different companies, in different institutions, etc. So seeing that things were moving in this direction, we also wanted to try and equip our investigators to play in this multi-modular space, if you will. So I think we're in an extremely interesting time as far as clinical research is concerned. Frankly, it had become a little sleepy for the last few decades, I would say. But now I think it's becoming really interesting. So we're obviously all interested in a shift from detection of large average effects to information that progressively is more relevant to individual patients. I mean, that's an ideal, but it's an ideal that at least is beginning to become tractable. So we can look at scale now in ways that we could never have done before. With increasing electronic health record coverage, we can ask questions or look for associations that we could never have dreamt of, no matter how large our practices were, and find unexpected disease associations, and with linked biobanks, begin to interrogate mechanism that might link these unexpected associations. But I think most importantly, besides those hypotheses generated at scale, we can now really use many deep phenotyping approaches to begin looking in relatively small numbers of people much more stringently with respect to the ways in which we address mechanistic hypotheses. And we've termed this approach human phenomic science. And importantly, that's complemented by approaches such as Mendelian randomization, and it's complemented by much more focused and creative approaches to trial design. So I think our ability to interrogate the human as a model system has really been enhanced over the last 5 years or so, I would say. Now, in terms of ITMAT, in terms of any organization, and many of you are responsible for many organizations, you're always asked, well, what's the impact? Where's the return on investment? How do we measure this? So we do what everybody else does, because we can. And this is just a picture of the translational connectome over the time course of our existence in Penn. So these are interdisciplinary, shared grants, but they could just as easily be patents or papers. And you can see that over the course of our existence, the connectome has become larger and much denser. But of course, the problem with this approach is there's no control group, right? So we actually don't know for sure how important we are in this evolution, although I assure you, depending on the audience, I can tell a different story as far as that's concerned. But I think more importantly, we've been around long enough that some actual discoveries have occurred. So we can look back and say, well, did we play a role, or what sort of role did we play in those actual discoveries? So here are some of the breakthroughs that people don't really quibble about that are sort of underway at the moment. As far as the ones in blue, we've had a fairly important role in Penn to play, and our investigators that we've supported through ITMAT played an important role, particularly in the CAR T-cell story, in the topical chemo for lymphoma, in much of the gene therapy work, particularly relevant to rare forms of blindness. So curing cancer, making the blind see, they're actually unequivocal accomplishments. They're not surrogates for accomplishment. And looking back, the story was very similar in all of these situations. A dedicated, focused investigator working for 10 or 20 years, not getting much help except being successful in getting grants from the NIH, and then getting to the point where things became ripe for translation into humans and not having anything like the right resource to be able to do that. And that's where we came in, along with other similar entities in-house, at a time when these investigators had no interest from industry, no interest from benefactors. For instance, Jean Bennett, who had a dog model of the eye disease that she subsequently cured with gene therapy. She had cured this dog model with gene therapy, and there was no interest at that time by industry, because that was before rare diseases became pervasively fashionable, as they are now. So there's no doubt that translational science can deliver, but the question is how precisely. So we're all very excited, for example, by checkpoint blockade now, but it works a lot of the time in vitro and some of the time in vivo. And the question is how? And how do we predict resistance? And how do we avoid resistance? And how do we detect the emergence of resistance? So these are really important and fundamental questions that, as the bandwagon keeps rolling, quite understandably, in terms of new applications, we have to give pause to. And they really rely on the types of skill sets and the human capital that I was talking about earlier in this lecture. And then, of course, the real challenge. How do we begin to put these novel, promising therapeutics that are often at the developmental stage, the early stage of application, in competing companies? How do we put them together? And as an audience of pharmacologists, you know the challenges of combinatorial drug therapy under any circumstances. And then there's the social issue. Are we just making drugs for people with extraordinarily good insurance or personal wealth? How do we spread the value of these discoveries in a way that is equitable in society? So I think these are the real challenges around translational science and its conversion into precision medicine. So I'm going to stray a little bit into the prostaglandin world. I saw a few people in the audience from that odd spot of the universe. If you think clinical pharmacology is an endangered species, you should try giving talks on prostaglandins. But I'm going to use it to illustrate some of the limitations of the current approach that we have. So larger isn't always better. So David alluded to the COX-2 inhibitor saga, and you'll remember that these drugs in the United States at the time of the withdrawal of Vioxx were earning around $7 billion a year in the US market. They were developed because they were less likely to cause serious GI adverse effects than traditional NSAIDs that inhibited both COX-1 and COX-2. And there were at least 2 large randomized trials that supported that contention. But in retrospect, the estimate was that around 3% of people in the US who got COX-2 inhibitors had actually suffered a GI adverse effect from an older NSAID. So direct-to-consumer advertising played a very big role in this story. Now, COX-1 and COX-2, 2 enzymes fundamental to making prostaglandins, are differentially regulated. But actually, the structures are almost superimposable. And one place where they differ is in the hydrophobic channel that allows the lipid substrate get to the active site, which is buried deep within the protein. In the case of COX-2, it is larger, it's more accommodating, there's a broader substrate specificity, and it has this side pocket, which doesn't exist in COX-1, which in retrospect afforded a structural explanation for the ability to develop NSAIDs that targeted specifically COX-2. And the 2 most famous of these were celecoxib and rofecoxib, Celebrex and Vioxx. And in this nice piece of work looking at that hydrophobic pocket, looking at celecoxib in magenta and rofecoxib in yellow, you don't need to be a structural biologist to see these 2 drugs sitting on top of each other, and you'd expect them to behave in a pretty similar way. So it turns out that they do. So the mechanism by which these drugs predispose to cardiovascular adverse events is by suppressing the formation of COX-2-dependent prostaglandins, particularly COX-2-dependent cardioprotective prostaglandins, particularly prostacyclin. And the fact that you don't need to invoke any additional mechanisms to explain that is illustrated in this slide, which shows the augmented thrombogenic response in response to either celecoxib or rofecoxib not being altered any further in the absence of the prostacyclin receptor. So both drugs will augment thrombogenesis compared to control, but there's no further augmentation by the drugs compared to the absence of the prostacyclin receptor, which itself augments thrombogenesis. So a lot of work is consistent with this story, and it's not surprising that the best clinical evidence that we have, which is an overview analysis of more than 750 randomized trials by Colin Bagent, shows very similar effects of both celecoxib and rofecoxib with respect to major cardiovascular events. This despite the fact that the regulatory response to the two drugs was widely divergent. So another observation that Colin made in his studies, which again was perhaps a little more controversial, was the suggestion that in the case of naproxen, that the likelihood of serious vascular events might be somewhat less than compared to other drugs. And of course, all of these data, on the one hand, are at scale, but on the other hand, are indirect comparisons. They're not direct randomized comparisons. They're indirect comparisons across studies. And the question is, how useful are these types of data? So with respect to the naproxen story, if naproxen is very long-lived, it sustains inhibition of COX-1-dependent thromboxane formation throughout the dosing interval. So effectively, it's a platelet inhibitor like low-dose aspirin. The problem in terms of deriving a message from Colin's data is that there's great heterogeneity in the kinetics of naproxen. So some people will have sustained steady-state concentrations throughout the dosing interval, and other people won't. And of course, dose is another factor as well. So even though there is this suggestion from the data at scale, we can't actually reduce that to a recommendation at the individual level without knowing something about the kinetics, without doing studies that look at outcomes based on factoring in the kinetics. So actually at the individual level, it has generated a lot of heat, but it's not actually very helpful to know that there's a sort of dilute aspirin effect of naproxen. Now, some time ago, we showed that there was actually a pharmacodynamic interaction between low-dose aspirin and non-steroidal anti-inflammatory drugs that inhibit COX-1 as well as COX-2. So these other drugs, aside from aspirin, are reversible active site, competitive active site inhibitors, whereas aspirin acetylates irreversibly a serine residue close to but not within the active site. And in a sense, for anucleate cells like platelets, paralyzes their action until new platelets are made. And this is just a sort of second round of that observation where now we're looking directly at Coxswain acetylation, the effect of aspirin action during chronic administration to healthy volunteers, and an indirect readout, which is formation of thromboxane. And what we're showing here is that this interaction is true for ibuprofen, which inhibits COX-1 and COX-2. It's true for naproxen, that inhibits COX-1 and COX-2. But this interaction is not present with a drug that just targets COX-2 because there's no COX-2 in platelets. So another example of going to scale recently have been two studies looking at celecoxib. So these are the Precision study and the Scott study. And between the two of them, they recruited roughly 30,000 people. And these were looking at non-inferiority, which is the way we look at comparative safety, crudely, in clinical trials. So again, they were looking at the comparison between naproxen, celecoxib, and ibuprofen. But the trials had multiple forms of bias that favored the declaration of non-inferiority with respect to celecoxib. As pharmacologists, you can appreciate the fact that if you use a less effective dose, you're probably likely to have less side effects. There was plenty of evidence that there were less efficacious doses used in these trials. There were correspondingly high and asymmetric dropout rates, reflecting mostly failure of analgesia. They favor celecoxib. Because it was difficult to recruit people into these studies, they relaxed the upper bound to 1.4. And if one looks at Colin Bajan's study, the dose of celecoxib that's used in these trials is indistinguishable from placebo with respect to cardiovascular risk, even though I've shown you that higher doses, which are more effective also, confer a cardiovascular risk in that data set. And finally, there was no randomization for aspirin usage or any objective measure of intake. So what we don't know, of course, is whether the declaration of non-inferiority reflects the attenuation of an asymmetric hazard from celecoxib due to the interference of the action of low-dose aspirin in the precision study in more than half of the patients with respect to cardioprotection. So large isn't always better. And who knows how we can move from this to try and get more useful information at the individual level. So this is a study we did many years ago where we looked at inter versus intra-individual variability of drug response, looking both at celecoxib and rofecoxib. And we used many assays with very low coefficients of variation, and we put the subjects through the same protocol 5 times. So we could actually measure inter versus intra-individual variability of drug response. And what that lets you calculate is the contribution to variability from fixed sources of variability, such as in your genome. So in these studies of healthy volunteers, if one had comprehensive information on genomic variation, which as we know we never have, the maximal estimate of its contribution to inter-individual variability was around 30%. So you then extrapolate that into a patient population, patients taking lots of drugs, older patients, and you get some sort of sense how for these common drugs that are used to treat syndromes like pain, how genomic information is going to be one-hand clapping in terms of variability. Then there are other sorts of things that have come out of the types of studies that we've been doing over the last several years. We've looked at dose-response relationships in cells, in model systems, in humans, comparing celecoxib and naproxen as two pharmacological probes. And what we found is quite striking. So this is the 2-minute introduction to the arachidonic acid biosynthetic pathway. That's it there in the middle, star of the show, and it's going in this direction to form prostaglandins. It's going in this direction to form leukotrienes and associated compounds, and in this direction to form P450-derived products. And the point is really that in magenta here are the genes that we showed clearly had a dose-dependent perturbation in just one of these systems in response to the drugs. So just looking at prostaglandins in this pathway is insufficient to capture the impact of drug effect in this pathway, never mind elsewhere. And clearly that can contribute to variability of drug response. Another issue is that we're all outbred. So if you look at the impact of different genetic backgrounds, in this case it's in mouse strains, we're doing something very similar in fish. And you look at the genes that we have identified and curated as cardinally relevant to this pathway. There's huge variability of expression as a function of genetic background, and it varies between tissues. So just to give you an example, there's COX-2 in adipose tissue varying more than 1,000-fold. So again, this hints at the variability of target expression never mind attendant genes, across tissues and across individuals. And indeed, just looking at transformed B lymphocytes ex vivo in healthy volunteers, we see a variability in expression of COX-2 of more than 200-fold. Does this matter in terms of modifying drug response? And they're amongst the things that we're interested in doing. So we have a large consortium called Pentacon, not Pentagon, that's trying to address this using these pharmacological probes in multiple systems, deep phenotyping people at the extremes of target expression, looking not just at the genome and the metabolome, but obviously at any other oome that you can think of, trying to identify genetic modifiers, and then using very diversified modeling approaches to try and develop predictive algorithms. And what we're using here is blood pressure as a quantitative surrogate of cardiovascular risk. We can obviously measure that in mice and humans, but also correspondingly in fish. And then if we can develop a predictive algorithm that actually works in terms of the very heterogeneous blood pressure response to NSAID exposure in humans, can we then validate that in terms of outcomes? So as far as this part of the talk is concerned, large and predictably slow randomized trials, in the case of Precision and Scott, have very effectively defended the franchise. While this was all going on, Celebrex sold $2 to $3 billion worth of drug per year, aided by aggressive direct-to-consumer advertising, saying, we don't know what caused all this, but we're doing the study to find out. And it took 11 years. And actually, as predicted before it started, Celebrex came off patent just as this trial concluded. There are multiple sources of bias in these trials favoring the non-inferiority of celecoxib, and they undermine the interpretability of the trials. And despite the inclusion of more than 30,000 people, they have provided no information of relevance to clinical practice. So the question is, can we go beyond that? And that's a bit like trying to land a tractor in Mars. And the hope is that we at least come out with some non-stick frying pans along the way. So I'm now going to switch to the less controversial part of this, and that is entrepreneurship and pricing. So we all know the stories. This audience for sure knows all the background of justification for drug pricing. It fosters innovation. So first of all, let's consider the promise, and there is a poster child, and that's Salvati, and I think it illustrates the argument quite nicely. So as you know, if you've Hep C, you've got a pretty good chance of cure with a 12-week course. The cost is a bit different in different countries, so there's tiered pricing, and where Hep C is a huge issue in Egypt, for example, the tiered pricing recognizes the asymmetry of our economies, and probably also the founder's national origin. But if all US patients were treated, we'd basically be using our drug budget just for one drug or for one condition. So, illustrative of that, you can see how the Medicare budget jumped from 2013 to 2014, never mind to 2015. But on the other hand, the counter-argument is that the company that developed this drug took an enormous risk and paid an enormous amount of money to get it, $11 billion. So you could say this is the way the system is supposed to work. The patent is being defended. It's been costed accordingly, and they have taken the risk, invested in the development of the drug, and look at the consequence in terms of patient lives. And the other part of the argument is, as more drugs came along, in a way, the system sort of worked the way it was supposed to. The payers began to consolidate. They began to harvest the value of the competition to push down the cost of drugs. So in a way, this is the poster child for the pricing structure that we presently have. Now, the other issue is this business of of how these precision medicines will be cost effective, and more importantly, how clearly they'll be shared. And of course, we're dealing with a lot of issues here that are very difficult to measure. So for example, if you look at this cost estimate of general support for patients with cystic fibrosis over their lifetime, it is somewhat, put in the shadow by the estimate of those who are likely to be responsive to one of the Kalydeco drugs or two of the Kalydeco drugs over their lifetime. But then the issue is the quality of life, the potential extension of life, those sorts of variables that are just so extraordinarily difficult to weigh. And I think a particular challenge is the gene therapy example in terms of pricing. So here we've had a big investment over a long period of time. We have an extraordinary curative promise over a range of diseases. We have a lot of companies that have IP in this area that have been valued very highly, despite the fact that they haven't actually produced very much. But here we're going to be dealing potentially with a one-and-done therapy that might be dramatically effective, but how do you price it? And often they're going to be focused on not just orphan diseases, but what we now call ultra-rare diseases. So I think there are really very fundamental, legitimate challenges around pricing that are very difficult to grapple with for good-willed people, no matter where they happen to find themselves in terms of structures. But there's another side to pricing and innovation, and that's the exploitation. So I updated my slides just before I came here because the judge wisely in New York refused the attempt to have a mistrial of this chap who before our last election was called the most hated man in America. He's now got a competitor, but this guy is the guy who raised the price of Daraprim by 5,000-fold. And this has become a bit of a common stunt these days. Take a cheap, effective drug that's been around for a very long time and jack up the price dramatically. And again, if you just look in the US example at the patients enrolled in Medicare, you can see the drug benefit cost 17% more than the previous year, even though the number of prescriptions increased only 3%. So clearly not a sustainable, not a politically sustainable situation. And, there are some examples that are just as egregious as the Daraprim. This South African company, for example, bought up busulfan and threatened to withdraw it from the Italian market unless the regulators accepted a 15,000% increase in price. But even our more conventional companies producing these breakthrough drugs, like checkpoint inhibitors that we're obviously extremely hopeful of, you can see taking these median cost of 8 cancer drugs across countries, the amounts of money are substantial and they have increased very rapidly over the last several years. And then there's this prospect of life extension. Not for us, for drugs. So a drug goes off patent, and actually one of the things that gets less attention in the United States is that now roughly 90% of the drugs prescribed are generic. And that can be obviously a very big deal. But in response to that, many of the companies we know and love deploy all sorts of games to try and keep alive their branded product that is now off patent and identical, or near as identical, as the generic competitor. And this just shows the branded price versus the generic price across a range of drugs. So this is the sort of behavior that obviously doesn't warm us to the public. And now there's a reaction beginning to take shape. So recently Maryland has passed a law on price gouging for off-patent meds to prevent increases that are not only unjustified but also legally unconscionable Interesting wording. So what is that? It's a relationship based on terms so egregiously unjust and so clearly tilted toward the party with superior bargaining power that no reasonable person would freely agree to them. And there's no upper limit on how much you can go after companies for that behavior. And this standard includes cases in which the seller vastly inflates the price of goods. So the Daraprim example would fall into this. What the Maryland legislature views as a non-competitive market, which they would then investigate, is one where there are 3 or fewer manufacturers of a compound, of a drug. And what they exempt are increases driven by price fluctuations for raw materials or active ingredients, difficulties in manufacturing or distribution, or changes in the international trade or tariffs. And of course, this is just Maryland, but this sort of thing has happened previously. It happened in Kentucky with generic substitution. And once passed in one state, it then spread to all of the states. So I think this is what's happening in terms of the reaction right now. There's another type of reaction, and I was actually quite interested in this paper that was recently in Nature Biotechnology. I spent 6 years doing Greek and Latin, and I'd never heard of magistral production of monoclonal antibodies, which probably says more about me than anything else. But basically, this is drug compounding on a local level by a licensed pharmacist producing drug for an individual named patient in response to a prescription by a physician. And the interesting thing in US terms about that is it may not only bypass FDA regulation, because nothing is crossing state lines, but also IP constraints, because it's for personal use. And these authors, who have been pursuing this for some time in Holland, suggest that from design to production in this system, they estimate costs way below current biopharmaceutical costings. So whether this is something wacky or this is something that has capacity to go to scale, we'll see. So I think we are left with this challenge. How do we foster innovation while containing cost and spreading benefit? So one thing we don't know in the States is the true cost to a patient. It's very opaque. There are headline costs, and then there are all sorts of discount deals that nobody actually knows much about except the parties designing the discount. I've been rude about our president. I'll continue to be rude about him. But I'll make one point, and that is that if you're a US consumer, you say, why should a Swiss consumer pay less for a drug by a Swiss multinational than I do sitting in the US, given that the GDP per head in Switzerland is considerably greater than in the US? You're used in the UK, thanks to Mike Rowlands' NICE and other maneuvers, this concept of reimbursement based on results. That's a very high hurdle within the US politically. But what we do need to move more towards is some sort of transparent evaluation of drug benefit. And then we need regulatory reform. I think initiatives to accelerate competition. I think we need to abolish pay for delay. I think we need to put an emphasis on less reward for first to market and more reward for information around how drugs act and parsing variability of response. We accelerate through the process. We need to hasten a little more slowly and gather more information. And finally, I'd like to turn to the altruistic sector, where I think there is a lesson for us in terms of considering IP reform. So as many of you know, there are outfits that have looked at the pricing issue in the altruistic sector, where intellectual property by definition is less of a cardinal driver in consideration. And this is just from the Drugs for Neglected Disease Initiative. And the estimates that they're making in terms of how they can either develop combinatorial therapies with existing drugs or how they can bring new drugs to market is way, way lower than the headline $4 billion that we have out for general consumption. So the issue is, can we translate the model of the altruistic sector into the for-profit sector with all the challenges that presents? But at least this shows that things can be done at least a lot more cheaply than $4 billion, which, as I said, builds in the cost of failure of a system that we know is quite imprecise. Now, IP can be extraordinarily valuable. I gave you the Salvati example, but in general sense, for biotech companies, IP is survival. So companies are placing patents and patent applications as collateral to raise money, and actually, again, the market is working the way that it should, in that many of these patent assets are released from securitization within a relatively short period of time. So clearly IP plays a very important role in the innovative process with which we're all familiar, and nobody could argue against that. But I think it is time to start thinking a little more radically about IP in this business. For example, in the case of Tesla, it's IP for free. The approach there is, let's give away the IP, and more people will drive these types of cars, and more of them will be ours. So in our business, IP is focused on the composition of matter. But the likelihood of that matter being an approved drug it's a long likelihood, right? Or a low likelihood. And in a sense, in the era of the large vertically integrated companies, focus on composition of matter didn't really matter. The chemist was in the company, but so was the clinical trialist. And if the drug got approved, everybody was going to do well. The shares would go up, everybody would do well. But now with the disintegration of these companies and a more modular approach to drug development, the question is how to engage those experts, those people you want to engage, that are not in your company, that are not in your country, to participate in the process. Well, to play in this space, you have to create a pot of gold. And the interesting thing about creating pots of gold to permit experimentation around IP and to permit the type of innovation that's occurred in the altruistic sector is that many different types of investors have created that type of resource. And people are beginning to use somewhat unconventional approaches. The dreaded credit default swaps are making a reappearance. Different approaches to try and reconsider our approach to intellectual property. Now, it turns out in this business, we're very used to modeling. We model all sorts of things. We model drug targets. We model biological networks. Drug development used to break down at PK for a long time until we got pretty good at modeling PK. We model market share. We model pricing. So one suggestion was to model the process. Before the process begins, to model where you think the greatest hurdles are going to occur, and to engage partners based on the prospective allocation of reward if the drug becomes approved. Now, obviously, you'll be accurate, inaccurate, but you can adjust for that as the process unfolds in a Bayesian fashion. And the attraction is that at the end of the day, you are postponing reward until value is actually realized. And of course, people will disagree, and you resolve that in the way you resolve disagreements now, and that's using the court system. So that's just one suggestion in terms of reconsidering the approach to IP. And this approach is going to be modeled shortly. And there are other fields where we're used to IP being around components, but the dominant IP being around the thing that you actually use that works. So I have some conclusions. You'd be glad I'm just about done in all sorts of ways. Drug development is risky and expensive, but life-altering breakthroughs continue to be made. If you create the infrastructure to allow academia to play in modular space, we believe that this will accelerate the process, ultimately decrease the cost, and increase efficiency. Larger trials are not always better. And now we're in the era to shift towards parsing variability in disease mechanisms and drug response. And if we can begin to do that, we will get more precise medication. Current pricing strategies are unsustainable and they're unfair. And it's time to question cost estimates of bringing a drug to market and to consider IP reform in drug development. So lots of the things I've talked about have drawn on the work of other people, in fact, virtually all of it. So I leave the last word to Leonard Cohen. The older I get, the surer I am that I'm not running the show. Thank you.